fructose, and fructose immediately becomes fructose 6-phosphate. So that fructose that was produced when sucrose was hydrolyzed jumps past these two steps here and gets right here. Okay, so that's why you may have seen more carbon dioxide produced in a shorter period of time, because you're actually speeding up the rate at which this reaction occurs. Which then leads to the actual question, well, why then do we need to consume glucose? Why shouldn't we consume larger amounts of fructose if we can make our glycolysis occur faster? The answer to that is metabolism. Apparently, when we consume fructose, it results in a larger quantity of fat cells being produced than when we consume glucose. All right, right back to it. So, fructose 6-phosphate, now we're going to use an enzyme called phosphofructokinase. Who wants to tell me what that enzyme does? What does it recognize, first of all?
It is phosphorus.
A phosphorylated form of
Fructose.
Good. Okay, and it's a kinase, so what do kinases do?
Transfer phosphorus groups.
Transfer of phosphate from one molecule to another. So, it recognizes fructose 6-phosphate. They're excited about it. And it transfers a phosphate onto that fructose 6-phosphate, giving us fructose 1,6-biphosphate. Again, called that because it's fructose. And this is the first carbon now. 1, 2, 3, 4, 5, 6. Okay? 1, 2, 3, 4, 5, 6. And we have a phosphate on the 1 carbon, on the 6 carbon, and there are 2 phosphates, so it's a biphosphate. Fructose 1,6-biphosphate. Now, while I might not require you to remember each and every intermediate, here is something I could do on the exam. I could give you a molecule that has been phosphorylated and ask you to name that molecule. So, not necessarily asking you to identify the difference between glucose and galactose, but I could give you a molecule that has a phosphate group on it and say, is this glucose 1, glucose 2, 3, 4, or 5 phosphate? And you would need to know how to do that. Okay. With glucose, the 1 carbon is always the farthest carbon on the right-hand side, right here. Okay, it's attached to this oxygen.
Can you say that one?
Sure. With glucose, the 1 carbon is always the one that's farthest on the right, attached to that oxygen in the ring structure. So this point here, where you've got the hydroxyl group going down, the hydrogen going up, that carbon, that's the 1 carbon on glucose. Because when this ring structure unravels, that carbon is actually the carbon that's part of the carbonyl group. And that makes it the one that's number 1 due to the functional group to which it's attached. Okay. All right. Any other questions? Yes, Gabe.
So just be able to recognize it to your phosphate that's on it?
Yes. In other words, I'm not going to give you 2 or 1 6-carbon ring structure like this and have you distinguish between glucose and galactose. That's not what we're here to do in this particular regard. Instead, I feel that the majority of this, what we're looking at really is the addition and the movement of those phosphate groups and how we name the molecule based upon it. Now, obviously, there are a bunch of other questions I could ask you about this, but in terms of naming molecules outside of the enzymes, I'd say that's one of the questions I can ask. Now, if I give you a molecule of, say, fructose 6-phosphate, and I asked you what enzyme would be responsible for adding another phosphate group to that, then I would expect you to know that it's phosphofructokinase. Because each and every one of these enzymes has a specific name that corresponds directly to its substrate. Yes?
I'm still confused about why fructose is called fructose 6 as opposed to, it's a, pentagon structure, so I guess there's 5 carbons around the side, so.
There are still technically 6 carbons in the molecule itself, though. So if you look here, here's carbon number 1, and is up here. Then this point here is carbon 2, carbon 3, carbon 4, carbon 5, and there's carbon 6.
Thank you.
So we closed the ring and made a pentagon, but now we actually have a carboxyl group, I believe. Actually, this is technically a carbon attached to a hydroxyl, and then another carbon attached to a hydroxyl group there, coming off of either of those two carbons, the 1 carbon and the 5 carbon. I'm sorry, the 2 carbon and the 5 carbon. So the 1 carbon in fructose is up here floating above the molecule, while in glucose it's actually part of the ring structure. Okay? Now, technically, I think where the confusion's coming is this would technically still be classified as, I believe fructose would still be classified as a hexose, because it does technically have 6 carbons, even though it's arranged in a pentagon shape.
Because if you said it, I was just talking about that, I didn't see what that's
Okay, tell you what, to do one better, I'll just go ahead and draw it on the board. All right. I have a marker open too long, so I try that. Okay, let's do a crash course in orgo. Okay, I'm going to leave the hydroxyl groups off because we don't need to worry about those so much right now. But I'm going to show you, this is CH, H, and then it should be CH. So there we go, okay. So this is fructose. Good so far? Okay. This is technically the 1 carbon on fructose. This represents the 2 carbon, 3, 4, 5, and the 6
Yeah,
carbon.
that's all I know. It's about, it was for the 6 months.
Yes, exactly.
Aside from the actual.
Right. And it is kind of confusing to go from, and I completely understand, because glucose is also a hexose, but it has a hexagon shape, and it makes sense. You know, you go 1, 2, 3, 4, 5, and then you have that final carbon up here, 6. That makes sense. This even still trips me up when I look at it sometimes. So, does that make sense to everybody? Everybody understands? Both of these also, remember, have the same molecular formula, C6H12O6. They're isomers, so. Okay. So, phosphofructokinase attaches another phosphate. We now have fructose 1,6-biphosphate. And we have this particular enzyme, aldolase, that comes in and results in the cleavage of this fructose 1,6-biphosphate. So remember, there were ketoses and aldoses we learned about at the very beginning when we were talking about carbon structures. Aldolase recognizes fructose, which would be classified as
An aldehyde.
right? Good. Okay. So we cleave it. Now we're going to wind up with one of 2 products. Dihydroxyacetone phosphate, or glyceraldehyde 3-phosphate. G3P is going to actually come back a little later when we start talking about photosynthesis. But again, whichever one of these we make, it doesn't matter. Glyceraldehyde 3-phosphate is the one we need to use to proceed. So if we do wind up making dihydroxyacetone phosphate, which we will invariably, we can use an enzyme called isomerase to alter its structure and give us glyceraldehyde 3-phosphate. Questions, concerns? You all seem very dead. I assume it's the content of this lecture that's doing it. Okay.
So after the aldolase, it produces the dihydroxyacetone phosphate.
That or the glyceraldehyde 3-phosphate. Basically, in cleaving this, right, because, well, actually it can create both, because if you look at the structure of fructose, it's a little different. Okay, so when we cleave it, one of the 2 products is going to be glyceraldehyde 3-phosphate, and one of the 2 products is going to be dihydroxyacetone phosphate. The isomerase can result in us switching between those products, so we can technically take glyceraldehyde 3-phosphate and turn it into dihydroxyacetone phosphate, or vice versa. But only glyceraldehyde 3-phosphate can be used to make pyruvate. So, after we make these products, whichever ones we wind up with, we're going to need to use isomerase to make glyceraldehyde 3-phosphate so we can then make pyruvate. This is all the energy investment that we're learning. 5-phosphate? Yes. Yes. If you break this apart and it produces both of these, this one can immediately be used in the next step. This one would have to be converted by isomerase to glyceraldehyde 3-phosphate before we would utilize it. Does everybody understand that? Okay. What we have done now is we have used energy investment to help us cleave a 6-carbon molecule into 2 3-carbon molecules. So the cleavage has occurred. The lytic part of glycolysis has occurred. Now what we need to do is convert both of these into pyruvate. Also remember, during glycolysis, we're producing two NADHs plus 2 protons. We're also producing, I guess technically 4 ATPs that go to 4 ATPs. But to do that, we first have to invest 2 ATPs. That gives us 2 ATPs. Okay? So, at this current point in time, what we have done so far is this, and we technically now have split the glucose into a 3-carbon sugar. Notice we haven't done any of this yet at all. This is only, sadly, the energy investment phase. All right. So now let's jump right on over and start talking about the energy payoff phase. All right, so what's going to happen in this first part of this phase is we're going to take the glyceraldehyde 3-phosphate. We're actually going to add a phosphate to it. We're going to do that by removing electrons from the molecule. We're going to remove those electrons by using a dehydrogenase. It's called triose phosphate dehydrogenase. Called that because it recognizes a 3-carbon sugar. It removes hydrogen, which remember represent the electrons, and in their place, it adds a phosphate. Okay? Yes, I can. It's called triose phosphate dehydrogenase because it recognizes a 3-carbon sugar. So that's the triose part. It removes hydrogens from our 3-carbon sugar. Remember, those hydrogens represent electrons.
Those are transferred to the ADH.
Exactly. They're transferred to the NAD plus, forming NADH, and those protons. And it then results in the addition of a phosphate group to the molecule.